First speaker is going to be Chuck Chan, who is an assistant professor in the Department of Surgery at Stanford University, and his research focuses on the biology of aging in stem cells and stem cell niches. Chuck?
Thank you, Bob. If you've ever seen Wolverine on a screen, regenerating from a major injury, or Benjamin Button aging backwards into a young Brad Pitt, you already understand the potential of therapeutic stem cell therapies. What I hope to do in the next few minutes is to give you a glimpse into the technical nitty-gritty that might make this movie magic not only feasible, but actually achievable in the next 10 years. Maybe less, if given the right type of support. So our lab's approach to stem cells was inspired partly by work from my PhD mentor, Irving Weissman, who, with Jerry Spangrud and new flow cytometry technology that was developed by the Herzenbergs here at Stanford, first led him to purify the first tissue-forming stem cell. And this is the hematopoietic stem cell, which at the single cell level has the ability to give rise to all of the cell types in the hematopoietic system. And the question is, how does it do that? Where are the regulatory signals coming from? So if you look at this map, actually what it shows this is actually the roadmap through which is multipotent hematopoietic stem cell can give rise to all of the cells of the hematopoietic system, and they are relatively different types of cells. Red blood cells, macrophages, B cells, they all come at some point from this hematopoietic stem cell. So how does it do that? So by actually purifying all these intermediates, then you have this roadmap. And then what is shown here are actually the regulatory signals that can guide this process. So our question is, what is the source of these regulatory signals? So if you think of the stem cells as the seed that gives rise to this hematopoietic tree of different types of blood cells, then our question is, what is the soil? And what is it made of? So this is looking at a cross-section of the bone marrow, and that's where the adult hematopoietic stem cells resides. This is a microscopic image of this bone marrow cavity, and you can see that it's actually made of many different types of cells. And so we established an assay whereby we transplant all of these different components into an area of the body that normally does not contain hematopoietic stem cells, and that is the kidney capsule. So we identify a population of blood vessel stem progenitor cells. And this is transplanted from a GFP mouse into a non-GFP kidney capsules. You can see it forms functional blood vessels. It carries blood, can rescue ischemia, but it does not have the ability to form a bone marrow cavity. So it does not make niche. We have identified a population of adipose progenitors, which when transplanted gives you adipocytes, but it also does not allow you to generate niche or bone marrow cavity. And lastly, we have identified a population of skeletal progenitors, which when transplanted into the kidney capsules, form a GFP piece of bones, recruit blood vessels from the kidney, and also within this bone marrow bone, there is a marrow cavity that forms, and it contains phenotypic and transplantable hematopoietic stem cells. So this shows that skeletal progenitors not only make bones, cartilage, but it also makes their microenvironment that forms, that supports a blood-forming stem cell, the hematopoietic stem cells. And with Michael Longacre, we teamed up together, and we found that this skeletal progenitor is actually made of a collection of individual skeletal progenitors, which came from a self-renewing skeletal stem cells. And this skeletal stem cells, similar to the hematopoietic system also gives rise to diverse cell types. In the case of the skeletal stem cells, it makes bone and cartilage, and also we see it gives rise to tendon cells. We identified the mouse skeletal stem cell in 2015 and the human skeletal stem cells in 2018. Now, in addition to identify these stem cells, the skeletal stem cell, what is important, as in the case of the hematopoietic system, we also lineage map this whole process. And now, by comparing the different types of intermediates, we have identified signals that can control the process of skeletal formation. So we found, for instance, that there is this one signal, BMP2, which has the ability to amplify skeletal stem cells. And then another signal, VEGF, has the ability to turn skeletal stem cells into bone lineages and tendon lineages. But if you block it and you apply BMP together, you can achieve something remarkable. You can make cartilage regenerate. This is really something because cartilage is the type of tissue that normally regenerates in adults. But in this case, we have taken off most of the cartilage off of a mouse knee. There's just a hole there. But when you apply these combinations of factor, BMP and VEGF inhibitor, you get stable blue formation of this blue staining cartilage. We also showed that this works in human tissues by transplanting human tissue, skeletal tissues into immunodeficient mice. And of course, this generated a lot of attention. And now we are at work applying these these cartilogenic factors in a large animal models in Yucatan pigs. And these same factors can regenerate a large cartilage defect introduced into the knee of a mouse. And without the factors, you see just white staining fibroblasts. With the factors, you see blue staining phenotypic chondrocytes. So I don't have time to go into the data for this, but we also took this approach and we applied it to the question of aging. And in aged individuals, as you may know, after a certain age, actually people don't die necessarily from, one of the major killers of people over a certain age is actually falls, and when their bones don't heal. And there's potentially many reasons for this, but we found one of the key reasons is the fact that the skeletal stem cells are aged. By working out this lineage map, we can compare young skeletal stem cells from aged skeletal stem cells, and we can find which signals are missing. And when we apply them together, as shown in this diagram, we can get the skeleton to regenerate back to the level of young adults. Moreover, we also showed that these combinations of factors can rejuvenate the bone marrow microenvironment so that they make more adaptive immune cell types and less pro-inflammatory cell types that can lead to inflammaging, which is also tied to atherosclerosis and Alzheimer's. So I'd be happy to discuss more of the details following up our research. But first, I want to thank our team and also our generous donors, and of course, the Tsai Human Performance Alliance. Thank you very much.